Let's start with a situation every peptide scientist has faced at least once. You run your sample through the LC, everything looks normal, and then suddenly an extra peak appears. It wasn't in your standard, it wasn't expected, and now you're left wondering what exactly is this? Today, we're going to walk through how LCMS helps us solve that mystery step by step, turning an unknown signal into a confirmed peptide identity. In peptide research and development, unexpected peaks are actually very common. Sometimes they come from synthesis impurities or side products. Sometimes stress testing creates degradation fragments. And sometimes, especially in biological samples like lysates or serum, the mixture is simply complex. The key point is, unknowns are inevitable. So instead of avoiding them, we need a reliable strategy to identify them. LCMS is powerful because it combines three capabilities into one workflow. First, liquid chromatography separates the mixture. Second, mass spectrometry tells us the molecular weight. And third, MSMS gives us sequence information. So instead of just seeing a peak, we can actually understand what molecule created it. That's why LCMS is considered the gold standard for peptide identification. Before we even touch the instrument, sample preparation is critical. We start by cleaning up the sample, desalting and filtering to remove particulates and salts. Then we use MS-compatible solvents like water, acetonitrile, and a small amount of formic acid. And we always avoid phosphate buffers because they suppress ionization and can clog the system. Think of it this way: clean samples give clean spectra. Most LCMS problems actually start here, not in the instrument. Next comes chromatographic separation. On a C18 column, peptides separate based on hydrophobicity. We usually run a gradient from 5% to 95% acetonitrile. This improves resolution and keeps peaks from overlapping. Our goal here is simple: isolate the mystery peak so the mass spectrometer sees only one peptide at a time. Good separation makes everything downstream easier. Now we look at the mass spectrum. Using electrospray ionization, peptides form multiple charge states, +2, +3, sometimes even higher. At first, the spectrum looks complicated, but deconvolution software combines those signals and reconstructs the true molecular weight. This gives us the peptide's molecular fingerprint. For example, we might discover our unknown has a mass of 1,456.72 daltons. Now we know exactly how big it is. That's our first big clue. Mass alone isn't enough, though. Different sequences can share similar weights. So we fragment the peptide. Using CID or HCD, the backbone breaks at amide bonds, creating smaller pieces called B ions and Y ions. These fragments form a ladder pattern. And each mass gap between fragments corresponds to a specific amino acid. It's almost like breaking a necklace bead by bead to figure out its order. Now we interpret those fragments. If the peptide comes from a known protein or library, we use database searching to match spectra to theoretical sequences. But if it's completely novel, we use de novo sequencing and rebuild the sequence from scratch. Finally, we confirm the result with high match scores and good sequence coverage, typically above 60%. That's when we can confidently say, yes, this is the peptide. Here are some practical lessons that make a big difference in real lab work. Calibrate your mass spectrometer daily to maintain high mass accuracy. Run internal peptide standards with every batch to verify performance. And always inspect the raw spectra manually. Never rely only on software. Because software can guess, but your scientific judgment confirms. Good habits save hours of troubleshooting later. So let's recap the journey. We started with a single unknown peak. We cleaned the sample, separated it with LC, measured its mass, fragmented it, decoded the sequence. And just like that, uncertainty became certainty. That's the beauty of LCMS. It doesn't just detect peptides, it tells their story. And now you know how to read it.